Hello, my name is Jason Murta. I'm an engineer with DPS Group, and I'm here to talk to you to give a brief introduction to oligonucleotide production and the most important factors involved in designing a facility to produce oligonucleotides. I'll give you some details of typical design issues that DPS have encountered when dealing with these sort of projects, and the different approaches possible depending on the individual requirements of each project. So, oligonucleotides are of great interest at the moment from a pharmaceutical perspective, due to their large potential for the likes of gene editing, gene silencing and the like, but their production process is also an interesting one. At the start of the process, a lot of organic solvents such as acetonitrile or toluene are used. Experience with handling these type of solvents may not be common in the biopharmaceutical industry, but the techniques required are commonplace elsewhere. Tank farm design, solvent distribution and control, fire protection guidelines. Use of solvents like these carries an inherent risk due to their flammability and potential for explosion, so there are strict guidelines in place regarding handling, storage and the design of facilities used to process these materials. As the process moves to the later stages, these solvents are replaced with aqueous solutions, and the unit operations become similar to those seen in the downstream processing section of the biopharmaceutical industry, with similar requirements for aseptic control against contamination. So, similar to the way that the oligonucleotide molecule itself is midway in size between the traditional small molecule from the API industry and the large molecule from the biopharma industry, the oligo process also lies somewhere between this traditional small molecule API and a large molecule biopharma industry, and involves process operations from both of these. The most unique feature of the production process is the synthesis skid, which is used to produce the oligonucleotides. This is effectively the heart of the process and entails a solid phase reaction process occurring within a packed column, where a number of reactions occur in sequence to enable a synthetic nucleotide to be added to the end of a chain of nucleotides attached to the solid phase. This sequence of reactions is repeated with different nucleotides until the complete oligonucleotide is formed, like sticking blocks of Lego on top of each other. A typical oligo would consist of about 20 of these blocks. The development work to achieve this arrangement has involved over 50 years of increasing levels of ingenuity from a chemistry perspective. But perhaps the most impressive thing about the results of all this very complex work is that the final product is an operation that is quite simple to operate. Once a suitable recipe has been developed for a product, it's a matter of connecting up all the supplies in the correct order and letting the skid take it from there. The major equipment items used for this process are of course the synthesizer skid, usually two chromatography skids for purification, typically a reverse phase and an ion exchange column, an ultrafiltration skid and probably a lyophilizer. Most of these operations require connections to a large number of vessels, possibly between 20 and 30. These are used for supplying solutions, reagents, buffers and inevitably waste collection. There are also a few reaction vessels required for the process with temperature and pressure control requirements. Batch size can vary depending on the scale of production. So the supply vessels can vary in volume from 50 litres up to 5,000 litres. The other equipment sizes don't change as dramatically with scale, with the synthesizer column diameter ranging from 10 centimeters to a maximum of possibly 1 meter, with a similar range for the chromatography columns. As I mentioned before, the rate of solvent usage is quite large. You can see from the graph the rate of increase in volume of the solvents used with the increase in batch size as indicated by the column diameter. The high usage of solvent as a proportion of the product produced is a concern within the oligonucleotide industry, and this is referred to as the MPI, the Mass Performance Index, which is the mass of solvent required in kilos to make 1 kilo of product. And as values can be typically up as high as 14,000, it can be seen why there is a focus at the moment on reducing this. Some other design considerations to take into account, which will vary depending on the scale of the facility, are the use of fixed or mobile vessels. Note, as a rule of thumb, once you go above 500 litres, you're not really mobile anymore. Mobile vessels have the advantage of flexibility and less hard pipework, but also require more manual intervention, whereas larger fixed vessels are more appropriate for a fixed, stable process. So the earlier the development stage of the process, the more useful a small, flexible process is, whereas at the later, more developed stages, a fixed, automated process is beneficial. Cleaning of the vessels should also be taken into account. Again, whether the vessels are fixed or mobile is important here, in determining what needs to be cleaned and how. Clean out of place COP stations are good for mobile vessels, but fixed vessels and their associated pipework require ideally automated validated cleaning processes to ensure cleanliness. Whether you're dealing with fixed or mobile vessels, hose or pipe, the process involves a lot of pipework and connections. See the picture in the slide for a typical comparison. So this must be taken into account during facility design. That picture is actually a 3D model that we used for a previous project, and the use of 3D modeling from an early stage is a great asset when you're designing a complex pipework system like this. Here we have a typical process flow for an oligonucleotide production process. Some of the individual steps may vary in their order, but this is generally what a process will look like. Once all the various solutions have been prepared for the synthesis reaction, the reaction is brought to completion, and at this stage the crude final product is still attached to the resin in the column. The product is then cleaved from this solid phase during the cleavage and deprotection step, which is normally done with aqueous ammonia, at which point the product is in solution and ready to be purified. A number of different chromatography techniques can be used for purification of the product. Reverse phase chromatography and ion exchange chromatography, as I said before, are the most common, but there are different techniques. After this, the product is typically in a dilute solution, possibly an aqueous saline solution, so an ultrafiltration skid can be used, both to remove salts and to concentrate the product down to a required concentration. In some cases, a white filmed evaporator may be used to increase the concentration even more if it's what's required. After this, the purified product can either be brought to the lyophilization stage if the desired final product is a single-stranded oligo, or if a double-stranded product is required, then it's transferred to the annealing process, where two single-stranded compounds are mixed together, heated, and allowed to combine into a double-stranded product. Once this annealing reaction is complete, the product is lyophilized, similarly to that for the single strand, and then formulated. The formulation process is typically to make a sterile injectable, but there's a lot of work going on in developing the optimum formulation process to improve bioavailability, so this might well change in the future. From a facility design perspective, some of the factors determining how the production areas are split can be seen above. Obviously at the start, with the high amounts of solvent present, sterility of the product is not a concern, so there are no cleanroom type requirements for the production areas at this point, although obviously they need to be able to handle large quantities of solvent, so explosion ratings and the like are important. A number of different chromatography techniques can be used for purification of the product, some of which involve the use of solvent and some of which are aqueous. Because this can vary, the chromatography and even the ultrafiltration skids are typically required to be capable of handling solvent, and the rooms they're in must also be. Although now, as the solvent begins to be replaced by aqueous solution, the risk of contamination becomes higher. So the requirement for the surrounding room quality gets increased to a grade D or an ISO 8. The annealing step similarly has a grade D requirement. Normally there is no solvent present at this stage, so the area could be classified as non-hazardous. But use of solvents is an option for some processes, so this classification may vary depending on the situation. The lyophilization and formulation steps, the cleanroom requirements, increase to a grade C at this point. Again, no issues with solvent reagents, etc. after this. Typical production surfaces required for the process. As mentioned previously, there's a lot of solvent handling. So solvent storage in a tank farm or some other suitable facility is required. Note that storage of large volumes of solvent on site can lead to the site requiring a higher safety rating. This depends on the country involved, and given the high levels of solvent required for oligonucleotide production, this is very likely to be necessary. Purified water is also required during the production process, so space for generation and distribution must be allowed for in the various rooms as required. The utilities and waste handling requirements for the facility should definitely be considered during the design phase, as they could be considerable. Due to the relatively high amounts of solvent waste generated, plans must be in place for handling all of this waste. The solvent waste streams involved can all be recovered and reused in solvent recovery units. Separate columns are required for acetonitrile and toluene recovery, if they have been segregated correctly when they were collected at first. Usually the biggest obstacle to utilising recovered solvent is that it involves a change to regulatory filing. So preemptive recognition in the filing of the possibility of using recovered solvent can save a huge amount of time and money. If off-site disposal or on-site incineration is required, a plan must also be in place for this. Waste storage, tankering, etc. Considerations must also be given to aqueous waste streams containing high COD solvent levels. Ultimately, the local environmental legislation and emission limits will determine the level of capital cost required here. Another consequence of the use of solvent is the requirement for nitrogen inertion of all vessels to minimise the risks of explosion. The use of nitrogen has its own hazards, asphyxiation, so the correct infrastructure, O2 monitoring, breathing air supplies and the like, and training are required to facilitate this. Further implications of solvent are the consequences of solvent emissions to the atmosphere. Although this would be dependent on the environmental legislation applicable for the site, it is likely that some sort of emission treatment is required to reduce the concentration of organic solvents admitted to the atmosphere. This could involve treatment ranging from the use of activated carbon beds, thermal oxidation, or cryogenic treatment, which, given the previously mentioned requirement for gaseous nitrogen and the likely requirement by the lyophilizer for liquid nitrogen, could be a good option for a new build. If there is a requirement for a capacity analysis, outline scheduling, etc. for a proposed process, or any query that requires a detailed analysis, DPS have developed a simulation of a typical oligonucleotide process. As you can see from the process flow diagram on the slide, it's not a simple one. Use of a simulation like this will allow checking that the equipment is appropriately sized, estimation of production throughputs, and a number of other applications. So that concludes my talk. For more information on oligonucleotide facilities, please go to www.dpsgroupglobal.com oligonucleotides.